Good afternoon, everyone, and who are tuning into today's Clinical Trials Science Forum. And it's a webinar and panel discussion. My name is Michael Roberts, and I'm Head of Investor Relations and Corporate Communications at Actinogen Medical. I'm very pleased to welcome you all to this afternoon's webinar, which will encompass presentation by three panelists, followed by a general discussion and question and answer session. It should last around 45 minutes, and that could stretch a little longer depending on how many questions we're getting. We will lodge a recording of the session as soon as possible after the conclusion, but allow about 24 hours for that to go up onto our Actinogen YouTube channel. We'll also advertise it on our social media platforms, and it'll also be on a under the presentations part of our website under the news tab. Our aim, as always, is to present our webinars in plain English. It's very technical sometimes, but we will get as close as we can to make it easy for you to understand. Please don't hesitate to ask a question if you don't understand something. Our panel this afternoon will be hosted by our Chief Medical Officer, Dr. Dana Hilt, who is joining us today from northeastern USA, where it's after 1 a.m. Dana will be joined on the panel by Professor John Harrison, who is an expert in clinical trials in cognition, and John's joining us from the UK, where it's eminently more acceptable 6 a.m. Welcome, John. And we're also joined by our clinical pharmacologist, Professor Paul Rolan, who's coming to us from Adelaide. So we're certainly an international company spread across wide geography. And I'd also like to note that our CEO, Stephen Goulet, is with us here this afternoon, and he's going to be moderating the Q&A session. Now, just move on. This is our typical usual disclaimer slide. Today's presentation might contain forward-looking statements, and I'm not going to read this out, obviously, but you can find it in other presentations on our website and in the presentation pack that was released today to the ASX. So just quickly, some housekeeping on Q&A. Many of you have been through this before. Just look for the Q&A icon at the bottom of the screen, type in your question, and then hit enter or the send icon. You can submit a question at any time during the presentation, and we may get answered. Steve will be keeping an eye on those, and we may address those as the presentation goes along or at the end when we're holding the specific Q&A section. We may choose to group like questions, and if we run out of time, we will reach out by email or post on our website. So our panel today is certainly an eminent one. Our first speaker and panel host is Dr. Dana C. Hilt, who has more than 25 years of drug development experience after a career as an academic neurologist caring for patients with neurodegenerative diseases, including Alzheimer's disease, and also teaching neurology. Dr. Hilt received his medical degree from Tufts University School of Medicine in Boston and trained in internal medicine at Harvard Medical School and neurology at the Johns Hopkins University Hospital before taking a position at the National Institutes of Health in Bethesda, Maryland. Dana has held numerous academic neurology positions, including conducting molecular biological research, taught clinical neurology and basic neurobiology, and cared for patients with neurodegenerative conditions such as Alzheimer's disease, Parkinson's disease, and ALS. He has extensive experience in phases 1 to 4 development of drugs for multiple neuropsychiatric conditions. And in addition, he's held senior clinical development and CMO positions at a number of pharmaceutical and biotech companies. And he served as our Chief Medical Officer at Actinogen for the past 1.5 years. Over to you, Dana.
Thank you very much, Michael, for the introduction. And for the listeners today, what the package that we'll be presenting is I will be giving a summary of what we know about Xanamem so far. We know that it enhances cognition, and we have 3 separate studies that have been conducted to date which show that. And we have 2 ongoing studies which we're investigating further the properties of Xanamem. And it's done so with excellent safety and tolerability. Then Professor Paul Rolan will be talking about some of the properties of Xanamem, which make it particularly suited to potentially be combined with other therapies or to treat a wide variety of patients with different conditions that are on a number of different medications. And then we'll finish the panel presentation portion with Professor John Harrison, who is really eminent and widely recognized expert in cognition and the clinical trials of neuropsychiatric conditions. John is a professor of neuropsychology and has multiple appointments at University College London in the UK, at the VU Medical Center in Amsterdam, and as the Chief Scientific and Development Officer of Scottish Brain Sciences. So I think we represent a fair amount of experience and a fair amount of insight into the measurement of cognition and neuropsychiatric and neurodegenerative diseases. So maybe we'll move to the first slide. So this is really the sort of top-line summary that I would like you to remember, that Xanamem is an oral once-a-day 11-beta-HSD1 isoform inhibitor, safe, well-tolerated, and has pro-cognitive effect. Well, what is 11-beta-HSD1? It is the enzyme which catalyzes the production of cortisol from cortisone. And so this blocks specifically in the brain the production of brain cortisol. And that turns out to be detrimental to people and is associated with decrease in cognitive function. So if we block the production of cortisol in the brain, it's a very reasonable expectation that we can have improvement in cognition and actually improvement in clinical function. Now, this approach has been validated in animal models of Alzheimer's disease. And as I mentioned, there are 3 separate previous trials that have been conducted today showing that it actually has pro-cognitive effects in both older, healthy, normal volunteers and Alzheimer's patients. It does so with an excellent safety profile, excellent tolerability, and a low likelihood of drug-drug interaction. And there's an ongoing phase 2b trial in Alzheimer's disease. And this follows an initial study which demonstrated pro-cognitive effects and indications of clinical benefit that I'll summarize in Alzheimer's disease. And then finally, Actinogen is conducting a study in patients with major depressive disorder and the cognitive impairment that is associated with that I'll tell you about. Next slide, please. And so this is just gives some more background about the 2 current clinical programs that Actinogen is conducting. Alzheimer's disease, suffice to say, and all of you listening, I'm sure, realize, is a tremendous problem. It's increasing as our population is aging. It's more than 30 million patients, and estimates are that it's going to be getting to 50 million patients worldwide in very short order, unfortunately. Interestingly, and something that you may not recognize, is that patients with major depressive disorder, about 60 to 70% of those patients have a significant cognitive deficit. And the average cognitive deficit of these patients is a half a standard deviation below normal. What does that mean? 1 standard deviation of cognition is 15 IQ points. So a half a standard deviation would be about 7 or 8 IQ points below normal. So this really impairs patients' function, and this cognitive impairment can persist even if the patient is responding to their antidepressant medication. Because Xanamem is not really dependent on amyloid or tau mechanism. It actually could be conceived to be potentially useful in a wide variety of conditions. And Dr. Harrison will be discussing some of those in a few minutes, and recognize that these represent, unfortunately, a very large population of patients that are not well treated today. Next slide, please. And so, as I mentioned, there are two studies that I'll go into in a little bit of detail. First of all is a study that's fully enrolled, and this study was conducted in Australia and the United Kingdom, patients with major depressive disorder. And we enrolled patients that are on background antidepressant medications or have previously had a good trial of them, but still have residual depression and still have residual cognitive impairment. The results of that trial will come out in early Q3 2024 this year. We've also, following on the initial study with Alzheimer's disease, it showed activity in those patients. We're doing a larger 220 patient phase 2B study, a 9-month study, and the interim analysis of that study, the results of the interim analysis will come out in mid-2025, the first 100 patients which are enrolled in Australia. Next, please. So as I've mentioned, and I've mentioned some of these points already, Xanamem is an oral low dose. We're exploring, really focusing on 10 milligrams right now, once a day treatment. And it's agnostic as to the presence of amyloid or tau. And so it could be conceived as a pro-cognitive drug that could be used widely. It gets into the brain, it reduces the cortisol production in the brain. And this can be hypothesized to lead to cognitive enhancement, which we've already shown in 3 separate studies, and even disease modification in Alzheimer's disease. And we have some data from our first study, which I'll share with you, that suggests that, in fact, we're starting to see that type of profile. I don't have time this evening to go into all of the data that suggests that blocking cortisol in brain may actually be an antidepressant effect. And so we're measuring depression in the ongoing depression study that we're doing. And not widely recognized is that this actually could enhance insulin sensitivity and may have anti-inflammatory effects in the brain. So there may be pleiotropic or multiple potential beneficial effects of this drug. Next, please. Now, what I'm showing here is the results in a schematic form of the antibodies to amyloid in Alzheimer's disease. On the y-axis is a measurement called the CDR sum of boxes. And this is a sort of a universal scale that's used in Alzheimer's disease progression studies to measure the clinical deficit in patients. And on the x-axis, is time. And so you've been hearing a lot in the literature and the public press recently about 2 or 3 antibodies that have actually shown a treatment effect in Alzheimer's disease. So the dotted line is the natural history. This is a progression of patients as over 18 months, they have a decrease in their function as represented by a 2.5 roughly point decrease in the CDR sum of boxes. So this is patients getting worse. And in the solid orange line, schematically is shown one of the antibodies, the antibody being developed by Lilly, donanemab, and that produces about a 0.5 point over 18 months difference. So that is a clinical difference, and that is an advance, that is a benefit. Lecanemab also produces about the same magnitude of benefit, and the one study that aducanumab did that did work also produces a benefit. So the real benefit, if you want to think about this, is the area between the solid line and the dotted line. That's really the clinical benefit for the patient. And that's pretty modest. And if you look above that, between the solid orange and the green line across the top, is the unmet medical need. Now, these antibodies quantitatively clear amyloid. So it's not likely that any antibody to amyloid is going to produce much different of a benefit or much greater benefit. And so the field has now recognized that meaningful treatment of patients with Alzheimer's disease cannot alone rely on antibody therapy, anti-amyloid antibody therapy. And as I like to say, as a physician that ran a clinic seeing these patients and treating patients with chronic disease, which is unfortunately what neurologic disease is, I've never seen a chronic disease that's treated with a single medicine. And so clearly there'll be need for additional medicines in our armamentarium to help these patients. And we think Xanamem could be one of those medications. Next, please. And this is data that's presently in press and we've published, and this is the analysis of the first study that Actinogen conducted in patients with mild Alzheimer's disease. And what we did in this study is we looked at patients that had elevated p-tau181. p-tau181 is a biomarker that one can measure in the blood. So you take a blood sample, you measure p-tau181, and if patients have clinical Alzheimer's disease, the early form MCI or early Alzheimer's disease, the diagnosis is unsure based on a clinical basis alone. So if you look at patients that have elevated p-tau181, it confirms really the diagnosis of Alzheimer's disease, but it also selects a patient population that is more likely to progress. And that's in fact what we saw in the first study. So in the individuals with elevated p-tau181, even over 3 months, the black solid line is the placebo group of patients showed a progression. And as you can see, the CDR sum of boxes on the left axis, they went down by about a point. Now, the patients that were treated with Xanamem, however, at 10 milligrams, you can see they had about a third of that as far as their progression. So, and even in this smaller pilot stage study, over a relatively short period of time, 3 months, there was a benefit, and this benefit was fairly substantial in its magnitude. And if you extrapolate this benefit over time, the other studies with the antibodies went for 18 months, 15 to 18 months, you can see that this would accrue a very substantial benefit. So that's really what we're doing in the next trial. We're exploring whether or not Xanamem can produce even greater benefit if one looks over a longer period of time. The next slide shows that in that study, not only was there a benefit in the clinical measure on the CDR sum of boxes, so if we move to the next one, please. On the left panel, I'm showing the patients in that study that had high p-tau. So these are the patients that most likely had Alzheimer's disease, certainly. And one can see the patients treated with Xanamem. What I'm measuring here is a cognition composite, measurement of thinking and cognition. And you can see that the Xanamem-treated patients didn't really progress over the observation period. They stayed the same. So that the confidence intervals, as we call it in the trade, overlap 0. They stayed stable. However, the placebo patients started to go downhill. Now, in the patients in that trial that had lower p-tau, so they either had very, very early Alzheimer's disease or maybe had some other condition, you can see that the Xanamem-treated patients had an improvement in cognition, and the placebo patients really didn't have that same improvement. So in this first Alzheimer's study, we have evidence then potential disease modification and clinical benefit in patients, and separately, measurements of procognitive or cognition enhancement or protection effects in patients with early Alzheimer's disease. And so we're now moving to a larger, longer study to confirm and extend these results. I'll talk a little bit about that. Next, please. And the drug was able to do this by having excellent safety and tolerability. I won't go into all the details, but there are a number of non-specific, if you will, adverse events that are seen in clinical trials. But you can see here from this table that patients treated with Xanamem and placebo had roughly equivalent frequency of having these adverse side effects, which are mild in nature. And importantly, when one does clinical trials, one is very attentive to something called a serious adverse event. This is something that could put the patient in the hospital, require some intervention. And they're really, to date, even though there are now between 400 and 500 patients treated, There's no treatment-related serious adverse events in the entire Xanamem development program. So the drug so far seems to have excellent safety and tolerability. Next slide. And so I mentioned the larger, longer trial in Alzheimer's disease, and that's shown schematically here. And we've started to enroll that in Australia. It's 10 milligrams of Xanamem or placebo, and it's going to be for 36 weeks observation period. The first 100 patients will be enrolled in Australia. And then what we'll do, what we call an interim analysis. We'll make an analysis of the results and see, well, what direction are we headed? Are we headed in a good direction? Does the drug have promise to have a positive trial over the full 220 patients over 36 weeks? And we're qualifying patients or making them eligible by assessing their p-tau levels. And they have to have a clinical diagnosis and elevation of p-tau. We're measuring cognition, and we've optimized a cognitive test battery that Dr. Harrison helped us develop or select using valid existing cognition measures. And we're also measuring the CDR sum of boxes, which I've already mentioned, and another activities of daily living or functional measure called the Amsterdam ADL scale. And that study is underway. Next slide. Then quickly to finish up my section, just a couple of slides on major depressive disorder. So this is not something that's widely recognized by people, that, as I mentioned, that patients with depression have cognitive impairment. And this is supported by a really large historic literature showing that elevated cortisol can be associated with the most severe forms of depression, that cortisol levels, if they're elevated, correlate with patient function, outcomes, relapse, and certainly cognitive function. This is widely prevalent in patients with MDD, major depressive disorder. Some studies show up to 80, even 90% of patients have these cognitive complaints, brain fog, difficulty making complex decisions. It can even impact their employment and day-to-day life functional level. And these cognitive symptoms can persist even if the patient is responding to antidepressant medications. So all of these data together, and you can look at this, of course, as Michael mentioned, we'll be supplying these slides and these references, really impair the patient's function. It's an unmet medical need. And so we decided to conduct a study in patients with major depressive disorder. And we already know, as I mentioned, and Dr. Rolan will comment on, there are two separate previous studies in normal older volunteers showing cognitive enhancement. I've already shown you the data from the initial Alzheimer's study showing that there is cognitive enhancement in that group of patients. Next slide. And so this is a study that is fully enrolled, and the data will be available early Q3. We're treating patients that have the diagnosis of major depressive disorder. They have a persistent depression, residual depression. As you know, depression is hard to treat. Patients cycle through multiple medications. The medications have significant side effects, and many patients don't tolerate them. And we're looking for patients that are on either a stable course of antidepressants or have been previously treated and are no longer taking them. As I mentioned, some patients don't tolerate them and still have a cognitive impairment of that half a standard deviation or 7 or 8 IQ point equivalent. And we're using as a primary measure a Cogstate cognitive test battery, which is a combination of paper and pencil, as well as a computerized battery, and looking at different aspects of cognitive function, and then secondarily looking at the universal scale to assess depression called the MADRS scale, and additional cognitive measures that Dr. Harrison helped us develop and to bring to this trial. Again, all of these are existing, valid, widely recognized and accepted measures of cognition. This study is conducted in Australia and the UK. We have 167 patients enrolled. Next slide. So, and finally, then in summary, that I've already mentioned that the major depressive disorder study, Xanamem as we call it, phase 2A trial, provide the top line data in early Q3. this year. And the drug could have both pro-cognitive effects and antidepressant effects. If it shows pro-cognitive effects, that'll be the fourth trial in which Xanamem has had a positive cognition benefit. And I think here that the cognition benefits can be extrapolated to and perhaps apply to Alzheimer's disease, but also other diseases, and Dr. Harrison will be talking about. In Alzheimer's disease, I've shown you some of the data from the initial trial, and I mentioned that's going to be published shortly. It's in press. And that trial, the trial we're now doing, the phase 2b trial, will be to confirm and extend those results, and the interim analysis will be in mid next year. Next slide. So that's a summary of sort of where the project is to date. And now I'd like to introduce Professor Paul Rolan, who is a clinical pharmacologist and has conducted literally hundreds of studies with experimental medications in patients, and is really a renowned expert in this area of assessing drug effects and how to measure them in patients, how to look for interactions with other medications, the metabolism, the distribution of drugs. And he's going to tell you more about Xanamem and some of the properties that it has that lends itself potentially to a number of different conditions. So, Dr. Rolan?
Well, thank you, Dana.
Staff to you.
Yes, so as mentioned, I'm going to describe some of the key properties of Xanamem that make it a highly attractive candidate to be a successful clinical medicine. Starting with the pharmacology and pharmacokinetics. Pharmacology, what's the intrinsic property of the drug? Well, we know that it interacts with the target enzyme at extremely low concentrations. And when we've looked at whether it interacts with other systems, and generally it doesn't. This mixture of high potency, that is, effects at low concentrations, and high selectivity means it's a very low likelihood for causing unwanted effects. And this will add to a better safety profile, and as Dana has mentioned, also reduces drug-drug interactions. In Alzheimer's disease, many of the elderly are on multiple medications, but also in the depression population, many of the licensed antidepressants interact adversely with pain medications, which are commonly taken in such a population, which is a big clinical problem. And we see Xanamem as being free from those liabilities. Regarding the pharmacokinetics, that's the concentrations of the drug. It's a very well-behaved drug which performs predictably. It's well absorbed. It's unaffected by food, so people can take it any time. And the half-life is around 14 hours, which means people can take it once a day, quite easy. We have measured the drug levels in the fluid in the brain. They're about 1/3 of those in plasma. That's a key issue for a drug which is meant to work in the brain, is whether it gets into the brain, because some drugs do not. We've also shown that the blood levels are consistent in young and elderly and Alzheimer's patients, making for a very easy and straightforward dosing regimen. And as mentioned, it's suitable for once daily dosing. Next slide, thanks. Now the pharmacodynamics. What does the drug do in the body? Well, as I'll show in the next couple of slides, we've already shown directly that the drug has high activity at the parts of the brain where we need it to work. And this is at the doses that we are using for the current clinical trials. Because the drug inhibits cortisol production in the brain, the key question is, does it inhibit cortisol production elsewhere? And the answer is clearly no. There is a counter-regulatory mechanism by which the cortisol-stimulating hormone ACTH has a modest increase within the normal level as a result of people taking Xanamem, and then we know that there is no sustained change in plasma cortisol levels during treatment, which is important because cortisol is an important hormone for health. Now, as Dana mentioned, we've seen cognitive improvement in 3 placebo-controlled trials. He's shown 1 in the Alzheimer's patients, and I'll go on to show those in the healthy older population. So next slide, thanks. And this is the brain scanning data. For those of you who are not familiar, there are 4 pictures here. The panel at the left, these are all pictures as if the brain has been horizontally sliced, and each slice is a picture there. And the brighter and warmer the color, the more activity, the more binding there is. The left-hand panel shows activity of the binding tracer in the absence of Xanamem. And you can see the brain lights up, and especially in the areas that are related to cognition, which I'll mention in the next slide. In the next 3 panels, you see the same picture, after 7 days of dosing with either 5, 10, or 20 milligrams of Xanamem. And at each of those dose levels, there is near complete attenuation of the binding. So this validates that our selection of the dosage range, so we know we are getting not only into the right target, but at the right dose level. If I can just show in the next slide, the regional brain activity. What this looks at is 4 different regions of the brain. This is the neocortex, where a lot of cognition takes place. Now, in this, on the vertical axis is binding, and the shaded box is the range of concentrations predicted to be effective in our target diseases based on the animal studies of around 50 to 60%. And we can see that in the cortex, both the purple bars on the cognitively normal and the orange bars for the Alzheimer's patients, where at all this dose range, even at 5 and 10 milligrams, we are substantially exceeding the concentrations necessary to reach the target effective concentration range. Next slide, thanks. This is just the data showing the cortisol levels in the Xanadu study that Dr. Hilt has mentioned. This is over the 12-week study, pre and post at various visits. The lighter bar is placebo, darker bar is Xanamem, and there is no effect on circulating plasma cortisol levels over this long-term study. Next slide, thanks. Now, these are the 2 studies showing improvements in cognition in healthy older participants. And in both of these 2 graphs, there is a measure of cognition as measured attention composite, 3 different tests of attention combined together. And the graphs are orientated, so up is better and down is clearly worse. The dotted boxes indicate the on-treatment period. The graph at the left was for our first study at 20 milligrams being treated for 12 weeks. And we can see that the Xanamem-treated participants in the black line had a steady improvement in attention over the study. A little paradoxically, there was a decrease in the placebo group, and that placebo group was quite small, only 12 participants, so that may not have been truly representative. But despite these small numbers, that was a statistically significant difference, showing improvement in cognition over that 12-week dosing period. However, as we wanted to concentrate on the lower dosing range, this is the Xanamem study, which is a shorter study of 6 weeks duration. We looked at both 5 and 10 milligrams with a similar attention composite. Here at 2 weeks, all treatment groups, including placebo, did improve, and that was consistent with a small learning effect that you sometimes see in such studies. However, those people on placebo did not improve further, had plateaued out. Yet those participants receiving Xanamem continued to improve without really a dose-response relationship. So these 2 studies together have shown a clinically relevant increase in cognition and confirmed that the dose range we now think is of therapeutic interest. So next slide. Summary. As I've mentioned, we know we're hitting the right target in the right part of the body. That's particularly important in drugs directed at the brain. We know it doesn't hit off-target systems, which would lead to reduced safety and drug interaction issues. There's a simple dosing regimen. As Dana has mentioned, with the hundreds of patients treated, the safety profile is remarkably good, and we know that we have studied it at the right dose. So thanks for your attention. I'm happy to answer any questions.
Thank you very much, Paul. It was a wonderful summary. Our final speaker is Professor John Harrison. I've already mentioned his affiliations, and he will be talking about the presence of cognitive impairments across the neuropsychiatric spectrum. John?
Thank you, Dr. Hilt, and thank you to Professor Rolan for setting the scene so nicely for me. I had just the one slide this morning or this afternoon, depending on where you are. And this I find incredibly helpful general sort of wallpaper for articulating really 2 themes. First of all, the nature and magnitude of any cognitive deficits in key disorders. You can see those listed in the first column. And across the top of the table are references to a number of cognitive domains. So these are, if you like, the atoms of cognition, parts of cognition, thinking, that we find it useful to sort of parcel up into individual domains. I'm going to focus very much on the first three lines, so major depression, bipolar, and schizophrenia. And also the last two lines, helpfully, we also have reference to Parkinson's disease and Alzheimer's disease. So in the time allotted to me, and I will be quite brief, I'm just going to talk about the nature of the deficits, which domains that we tend to focus on as presenting features for the different indications, and some reference also to test selection. So a good place to start always is with cognitive domains. There are a number listed across the top of the table. This is sort of academic thoroughness. We're here accommodating all tastes and flavors. There are some musical differences, I think, in the community about which are the key domains. I'm going to focus very much on the idea that what we should be most concerned with are issues of attention, concentration in lay language, episodic memory, just memory as we would refer to it, executive function, so capacity to organize, plan, think ahead, and strategize, and working memory, which we'll think of very much as the space in your cognition where you solve problems. So really, I think of most of the action in cognition as being in those four domains. And if I take you very quickly through the 5 areas of interest, let's start with major depression. So as Dr. Hilt very helpfully pointed out earlier, a number of patients who even are between episodes of mood disorder still report some significant cognitive difficulty, and often that's translated into functional difficulty. We may get into that distinction in the Q&A, but the idea that cognition is the substrate of function is, I think, a very well-established one. So what we see is modest but clinically relevant levels of impairment across a number of domains. So bipolar disorder, the pattern is very similar. What tends to be at variance is that the profundity of the deficits that present seem to be that little bit greater. So much the same areas of cognition, but the magnitude of the deficit is typically reported to be higher. Schizophrenia, which remains a serious area of unmet need, as indeed do all the indications in this table. This is a disorder which afflicts often people in the very early stages of their life, can be significantly debilitating, and the therapies that we currently have tend to be very focused on the so-called negative signs and symptoms, the positive signs and symptoms, excuse me. So the hallucinations, delusions, much of that leaves the core deficits of cognition untouched. And as you can see, the profundity and breadth of those deficits in schizophrenia is very significant. So again, a clear area of unmet need. If I jump to the bottom of the table, we have Alzheimer's disease. You've already heard a good deal about this. We tend to think of Alzheimer's disease as primarily a disorder of memory, and often that's the presenting sign or symptom of people coming to see us in clinic. Really important to remember that it's actually a generalized disorder of cognition. So in clinical drug trials to date, we have been very focused on the idea of looking at episodic memory, your recollection of new information. In fact, we are aware and see routinely in clinic people who present with memories that aren't great, but their presenting feature that is most debilitating is one of concentration, one of problem solving, or one of organizing their thinking. Most clinical trials in Alzheimer's disease pretty much omit measurement of anything other than episodic memory. And the community at large, and including agencies, particularly the FDA in the last few years, has highlighted this as being an important area that we need to fix. It's reassuring to see in this study that the breadth of assessment in the Alzheimer's and the depression arms of the program are very focused on a broad assessment of cognition. And lastly, just wanted to highlight that in Parkinson's disease, which we tend to think of as primarily as a disorder of movement, it's not uncommon that as many as a third of patients presenting in de novo, so just after diagnosis, often have a detectable and conspicuous deficit in cognition. Sadly, as the disease progresses, that's often become more widespread, and by the end stage of the disease, it's quite unusual to find patients that have no cognitive deficit. So I feel very strongly that we've overlooked in Parkinson's disease the cognitive component, and that's unfortunate because often when one speaks to caregivers, to clinicians, and to the patients living with the disease themselves, they'll often report that for them, this is one of the key debilitating aspects of the disease. So lots of opportunity. Major depressive disorder and Alzheimer's disease are clearly front and center for Actinogen. At that point, I'm going to leave the slide here and pass back to Michael. Thank you, everybody.
Thanks for that, John. We'll come back to this slide, but just quickly, a reminder of the Q&A procedure. We've already got a couple there, but we'd love to see more questions. Just look for the Q&A icon at the bottom of the screen, type your question, and send it to us. So I'm going to throw back to Dana here, but we'll just start the panel discussion with this slide over here, Dana.
Thank you, Michael. So I thought we would start some discussion amongst the group looking at this, and John's already made the point, but I might ask him to comment further on the ubiquity of this. If one looks on the left side, one sees almost the table of contents of a neuropsychiatric textbook. And these conditions are really widespread, and they do impact the patient's function. And the patients, if you look at these different conditions, are on a wide variety of neuroactive drugs, which do have side effects, tolerability issues. So maybe Dr. Harrison, John, you could comment on what the present therapies actually produce in the patients as far as both side effects and do they really robustly treat the cognitive impairment in these patients?
Yeah, no, thank you, Dana. And I think in response to that, I'll focus very much on major depressive disorder and Alzheimer's disease. So if I think about Alzheimer's disease in particular, for donanemab, for lecanemab, for all the currently under consideration or maybe even approved in the US treatments, it's very much that we understand that there's an impact on episodic memory. We rather suspect that these treatments also have beneficial effects in other areas of cognition. But as the slides that you presented earlier clearly illustrate, these aren't a fix for the disorder. And the community in Alzheimer's disease is very fixed on the idea that idea of bringing you back to normal levels of cognition is almost certainly going to require multiple targets. So we might be able to slightly arrest the progression with the use of disease-modifying therapies like donanemab. There's still plenty of room to try to get you back to normal levels of cognition with symptomatic relieving drugs, of which Xanamem looks like a very good candidate. So looking within Alzheimer's disease, I think we might reasonably say that there's the prospect of arresting progression and the symptomatic relieving drugs rescuing your established deficits in memory, attention, working memory, and executive function. So lots of room for still meeting that unmet need. In major depressive disorder, our experience of looking at this, and I'm thinking particularly of trials that I've been associated with, there was, I think, 10 years ago, this idea that the cognitive deficits consequent on major depressive disorder were in some strong sense related to the elevation or the reduction in mood. I think we know now that actually they're different core deficits. So work of the kind that was done with Lundbeck's drug, the Brintellix, strongly suggested that there were core deficits of cognition and of mood, but they could vary across patients. Some patients responded in terms of mood, but not on cognition, and vice versa. The idea that we would try to fix attention and working memory, I think, is a very admirable proposition, not least because most of what we do starts with attention. The first stage of remembering something is paying attention to it. Maintaining the contents of working memory when you're problem solving requires you to pay attention. So I think there's lots of room for improvement. And within the context of drug development, disease-modifying therapies certainly have a place, but the symptomatic relieving component still represents a clear and obvious target for us.
Thank you, John. And you didn't mention in your discussion such things as PTSD or schizophrenia. I think schizophrenia is a condition that is recognized as very debilitating, but people don't recognize the cognitive impairment. And that's pretty significant, isn't it, John?
It is, Dana, thank you. And in fact, there has been in the last few years a very substantial movement within the community to highlight just this. So the Matrix initiative that was instituted in the US has focused on a number of domains. If people look across the line of schizophrenia, the M that's attached to each of those domains illustrates domains that the Matrix groups have asked us to focus on. And the idea of cognitive impairment associated with schizophrenia, CIAS, has for many sponsors been a clear target. And when one finds a pro-cognitive drug, as we strongly suspect Xanamem will prove to be, it's always tempting to look to other indications in which there's significant
Yes.
cognitive unmet need.
Thank you, John. A quick question for Paul. Paul, you mentioned some of the properties of Xanamem that could lend itself to be widely applicable to different conditions. I know that in your experience as a physician, not only if you have wide experience and deep experience in clinical pharmacology and looking at experimental drugs, but you also treated patients with pain syndromes, which were always treated with a variety of neuroactive compounds, which have side effects, sometimes poorly tolerated. Maybe you could elaborate for our listeners a little bit on some of the potential side effects that these drugs have, and the fact that Xanamem appears to be well tolerated and could be added onto the drugs that the patients on this list of these conditions, the drugs that these patients have to take.
Yes, there is a very large number of therapeutics out for neurological and CNS disease, but many of them work with subtypes of the serotonin system. And unfortunately, that's where a lot of interactions, which can be quite serious, can occur. So I mentioned the most popular type of antidepressants, the serotonin selective reuptake inhibitors, interact with a number of pain medications such as tramadol, and also drugs. We also use other antidepressants such as amitriptyline. So in clinical practice, since pain and depression happen so commonly together, we're often limited in safely treating people. What's particularly attractive about Xanamem, it's completely different. It doesn't interact with the serotonin system. It's working on this cortisol system. And we've seen no evidence of any system interactions as a result of Xanamem. So whether people have concomitant depression or other mood disorders or PTSD, schizophrenia, for example, we can't really see that there's going to be any of these difficulties one faces in clinical management, because sometimes in clinical practice you're having to choose between the devil and the deep blue sea, whether you can prioritize treatment for one condition over another, or you may have to go off-label using drugs which are recommended to be used with extreme caution when the clinical situation requires it. So that's complicated and generally best left to specialists, whereas these drugs, these conditions are so common, we need treatments that are user-friendly and can be given by a family physician. So as you indicated, that with Xanamem, we can't see any reason why this is a drug that cannot be given safely and easily by a family physician for the range of conditions that we see. Whereas for people with multiple simultaneous conditions, that's often the area which is quite tricky to navigate for the generalist.
Yes, also as a clinician treating patients, I found juggling different medications, as I mentioned, in chronic diseases, patients almost axiomatically require multiple medications. It's always a huge challenge, both for the physician, but even more importantly for the patient, because these drugs that we use to treat these conditions as best we can do have a lot of side effects, and one has to be very careful about the doses. And sometimes there are drug-drug interactions, which are really vexing for the patient and for the physician. So it's clear that all the diseases on this list are not optimally treated. And just to amplify one of the points that John made, when I was a clinician practicing and teaching neurology, I treated patients with Alzheimer's disease and Parkinson's disease. And it's not widely recognized, for example, as John mentioned, in Parkinson's disease, that you have cognitive impairment. Most patients with Parkinson's disease, as they progress through their illness and they get to the later stages of the condition, do express cognitive impairment. And that is one of the reasons that patients really have to go into extended care situations, or really have problems staying in their home and functioning. So cognitive impairment across all these conditions is a really debilitating aspect of these diseases. So I think we've went on for quite some time discussing, and so maybe I'll ask.
Yes. I'll stop sharing, Dana, and so everyone can see the panel. And there's quite a few questions in the queue, so Steve will moderate that with yourself.
Thanks, and for those who need to leave at this time point, we will be, as Michael said, putting this recording up on the website and our YouTube channel in due course within the next 24 hours. So if you do have to leave, we will be answering the questions in the next 10 or 15 minutes. So There are some very good questions here. Hopefully we've got time for some relatively quick answers. So to get to all of them, we have about 8 or 9.
Yes,
The first question is a clinical one. Would it be beneficial, the meaning Xanamem, for insomnia, from an anonymous attendee? Perhaps you might follow some appropriate patients already being treated to see if it is, perhaps using Fitbit or an Apple Watch.
I could start, or Paul, maybe you could comment on that.
We actually did look in the studies. Of course, we do look for a broad range of effects. So we have actually looked at sleep quality in some of the earlier studies, and we saw no beneficial nor negative effect. And that's important because, again, some of the treatments for depression, when you start treating, people get agitation and insomnia. So we've got, if you like, a clean profile. We don't think it really affects that. But you're absolutely right. The question that the Apple Watch or Fitbit, they're excellent ways of tracking sleep quality. We didn't use that, but we just used validated questionnaires that assess sleep quality, and we saw no signal for either beneficial or negative effects.
Thanks,
Yeah,
Paul.
just make a quick additional comment that the use of actigraphy, which the Apple Watch, Fitbit, and so forth are simple examples of, is really looming large in Parkinson's disease, and that's being used more and more as a clinical measure in patients.
Yeah. The next question I'll tackle, which is a sort of a drug development question. If these phase 2A trial in cognition and depression and the phase 2B in Alzheimer's are 100% successful, what are the next steps and when would the drug get to market in the best case? So look, the easiest way to answer that is that obviously there's a number of clinical trials, which the FDA and other regulators call pivotal trials, which we talk to them about, design with them. Those need to be conducted, typically with a small molecule. Despite the good-looking safety profile, the FDA wouldn't generally regard 1,500 patients treated, at least 100 patients treated for 12 months as the minimum safety database requirement. So that will take a few years. We don't comment on specific timelines for trials that are several years away. But on our website, we do have a very good analyst report from our analyst, Puya Hermani, who estimates that we might be able to get the drug approved in 2028 for depression and 2029 for Alzheimer's disease. So it's in the order of that timeline. The next question is, what percentage of Alzheimer's patients have elevated p-tau biomarker levels?
Maybe I can tackle that. So if you talk about patients that have clear Alzheimer's disease, they have cognitive impairment and some functional deficit, the majority of those patients are going to have elevated p-tau. In fact, the National Institutes of Aging and the AAIC, the Alzheimer's Association, have issued new guidelines suggesting that a clinical diagnosis and an elevation in a blood biomarker, which p-tau is really the leading blood biomarker for this characterization, will be the new format eventually to diagnose the disease. Right now, you have a clinical diagnosis and you have to have a PET scan, which is very expensive, invasive, $5,000 procedure, or have lumbar puncture and CSF analysis, which is also invasive and expensive. So p-tau elevation is pretty universal in patients with Alzheimer's disease.
Thanks, Dana. Peter asked, how many of the planned 220 patients in the Alzheimer's trial have been recruited?
So we're at the initial stages of enrollment, and in the clinical trial, it takes a while to get rolling. Once you get rolling, then the patients come in rapid succession, if you will. So we're starting the trial in Australia. So we're in the early phases of enrollment. We don't comment on exact numbers of patients enrolled at any particular trial, but we have more than 50% of the sites, 60% of the sites activated, and they're screening and enrolling patients. Of all the centers that I've talked to, and I've talked to a number of centers in Australia as well as other countries, this particular trial is very attractive to these centers because most of the drugs in development for Alzheimer's disease are really looking at MCI, very, very early Alzheimer's patients, and there's really nothing there for patients who actually have symptoms and have the disease. So sites are very enthusiastic about seeing if their patients can enroll in such a study.
Thanks, Dana. Brendan asked, could you comment further on how the Xanamem results in the next quarter could be extrapolated as a preview for the possible results? I'm assuming he's referring to the endpoints we've chosen, such as cognition or CDR sum of boxes in Alzheimer's.
Maybe I can start, or John, you could comment on that.
Yeah, thanks, Dan. I will, and I'll be brief. So what I would just point out is that when we've tried to do this in the past, so the predictive validity of results in one indication into another indication, the vortioxetine example I mentioned is probably a really good example of that. So we established the pro-cognitive effect of treatment in major depressive disorder. Subsequent studies that have been conducted, typically investigator-led studies of early Alzheimer's disease, have replicated the kind of cognitive benefits that we saw in the depression study. So there's certainly precedent for that expectation.
Great, thank you very much. Cameron was asking a question which I'm not 100% sure I fully understand. He was a bit confused about the healthy older volunteer data. And he was talking about a crossover between placebo and 10 milligrams of Xanamem. I think he might be talking about the fact that the 5 and 10 milligram data in the Xanamem study, Paul, that you presented, sort of went down again at the end in one case, and one went one way and one went the other. Is that, are you worried about that?
No, remember, these are relatively small samples, only about 30 participants in each treatment group. These are really part of an exploratory program looking for signal detection. And so one expects to see some variability. But what we see is between 5 and 10 milligrams, there's a consistent pattern of improvement over a placebo. And you'll get smoother profiles when you go to studies with significantly larger numbers.
Yeah, thank you. James asked, is it possible, this is a great question, to seek FDA approval for general cognitive improvement or enhancement as opposed to targeting specific diseases?
I'll take that one. The short answer is no. That basically the FDA takes the position that they're in the business of approving drugs for recognized and defined diseases. And so you'll see that on Dr. Harrison's slide, on John's slide, all of those are recognized and valid, if you will, diseases. So they would consider drugs developed for those conditions to be something they'd be interested in and would work with us on, but not for use in the general population. Now, there are drugs that are approved for particular conditions, which sometimes are used in the general population. So that does occur, but that's not what the FDA is regulating us on, and the EMA and the other PGA and other regulatory bodies.
But it's not beyond the realms of possibility in the future. As Dana says, you can market a drug for a disease. Now, there are some drugs that are approved for a symptom. So, for example, pain is one of those things because it's so well characterized. But there are some drugs which are only approved for pain due to neuropathic disease, for example. And so you have to, that may be only that symptom associated with that disease. So it's a possibility. That comes back to our pipeline in a pill. We have to take it piecemeal. What's the old thing about how do you eat an elephant? The answer is one bite at a time. So that might be where we actually end up going, and that would be a wonderful outcome, but we would have to get it through one bite at a time. But if, for example, we can see improvement in cognition, in schizophrenia, and in depression, and then after a bite, you can actually say there as well, there's a lot of blur in some of those conditions. So that's something where it could go, but that would be very optimistic and long term. Not yet.
And then I'm conscious of time, so I'll just take one more question before I hand it before we close. And Maurice asks a great question that I get a lot as the CEO. Is the main goal of the company to present the results of these trials to big pharma, and how do you see the prospect of a joint venture, and if so, are you looking at a particular timeline? So in this type of drug, in the drug development world, smaller companies like Actinogen are always talking to potential partners, both big pharma partners and smaller and regional partners. There's a lot of interest in our programs. We will be very busy presenting the results of the Xanamem study next quarter to a number of companies that are interested, not just in depression, but in the possibility that this drug could be used in schizophrenia. Obviously, sure, the positive results there would further improve the likelihood of success of the Alzheimer's trial, and the possibility this could be used then beyond Alzheimer's in Parkinson's, in the other types of dementia that we saw mentioned by John. And so the timing of that, it always takes at least a few months to do a deal. Companies like Neuron did a very successful deal with Acadia for their initial molecule and are reaping the rewards now with large milestone payments and royalties. That's the sort of deal that we would contemplate, but it's very hard to put a timeline on it because it depends how good the results are. It depends on the appetite for doing transactions. And obviously, we are very focused on delivering the maximum shareholder value within a reasonable period of time. So in the next couple of years, we would like to see a fruitful partnership where we can have more resources brought to the table to develop this drug in multiple indications and bring it to patients as soon as possible.
I think that's brought us to the end of the session. Certainly, we've spent an hour, and it's been fantastic. So thanks to the panel, Dana and Paul and John for your presentations and discussion and answering questions. To everyone online, feel free to reach out afterwards. As I say, the recording will be up as soon as we can, and reach out. The contact details are on the last slide in today's pack, and I'll just ask Steve to wrap up the afternoon.
Thanks, Michael. So hopefully we've been able to explain that we've got a lot of great clinical data showing that the drug is pro-cognitive. It's got great clinical safety. It's the potential to combine it with other medications, and in multiple diseases where cognition impairment is a major problem. It's certainly a very good-looking candidate for that. And we're looking forward to having near-term confirmatory clinical results in our cognitive impairment and depression trial in just a few months' time. So, and thank you again to all the panelists and to all of those who attended today.
Thanks, everyone. See you again soon.
Thank you.